is Caroline. Just a brief introduction. I'm a researcher at Stellenbosch University. I work specifically on tuberculosis, and my interests are in whole organ imaging. So I use tissue clarification, and I do this on mouse lung samples so we can image TB disease in 3D. And so I have a lot of experience with fluorescent labeling and sample preparation, and I do the honors course for microscopy. So we're a little bit out of order. Unfortunately, this was my fault as I couldn't present last week. I was away. So I know some of this will already be familiar to you. I know the fluorescent course has already been done, but I hope this will be an interesting talk. And I think this is an important one. Sample preparation. Sorry, I'm not able to move forward with my arrows. Here we go. Sample preparation really is kind of a key step, and I really think probably one of the most important steps. So by the end of this lecture, what I'm hoping is that there will be a greater understanding for the need for appropriate sample preparation and why it's so important, be able to choose between different fixation methods for different sample types, understand how and why permeabilisation should be used, have a brief overview of the different labeling protocols and which ones will be applicable to what you're trying to achieve, and then select the appropriate labeling strategy for different applications. So as I mentioned, sample preparation really, in my opinion, is the most important step in your microscopy experiment. You can always restain a different section, but you will never be able to store and properly fix a sample again. So once you acquire it and you prepare it, that's kind of the end step for that. So as we know, especially in the medical and biological research, we need to understand firstly a normal structure and function at different scales. Of course, organisms are made up of different organ systems, and organs are made up of tissues, which are made up of cells and ultimately are comprised of organelles. And so having a proper understanding of how these look at different scales is really important. And so many diseases are characterized because they have these alterations from the normal state. And oftentimes these can only be seen at the microscopic level. So proper sample preparation, where you take this into account, is really important. If you want to completely understand an organism and understand disease processes, you need to minimize the amount of distortion and ensure that you really have an accurate representation of this state. And so oftentimes sample preparation, if done badly, can introduce aberrations that really kind of compromise the experiment from the beginning, and you won't be able to get a good output from the experiment. So you want to ensure that you have high quality images that really provide the best sample so that you can ask the correct questions. So there are a number of requirements for a specimen in terms of your sample preparation. And the first, which we've already mentioned, is that it must be preserved in a lifelike state. You really want to represent as much as possible the real scenario. And everything that you do to your sample can introduces different types of aberrations. So it's really important to understand how fixation affects your sample, how the preservation strategy, cutting even, the sample orientation, all of these are things that should always be in the back of your mind when you're preparing your sample. And you want to try and preserve the lifelike state of your sample. The sample should ideally be transparent. Of course, there's new strategies now in terms of tissue clarification, but that's really kind of getting into the weeds. Ultimately, your sample needs to be transparent so that light can pass through it. And tied to this is that your sample should be thin and flat. So ideally, you don't want light distorted, especially for fluorescence. This is quite important. Light should be able to pass kind of in a homogeneous way through the sample. And finally, you need good contrast. Of course, there are different things like phase contrast, and we'll talk about that in a little bit, but you need to have differentially colored components in your sample so that you can distinguish between each of them. So these are the kind of the key requirements. Of course, there's other things to take into consideration, which we'll touch on in a minute. So how do we achieve contrast? The first is really kind of the simplest, which is label free. And you can do this via phase contrast, dark field, or polarized lights. And so for each of these, we've provided an example. So for phase contrast, something like Plasmodia or trypanosoma or bacteria can be visualized in this way. And this is by no means an exhaustive list. You can, of course, do histology stains, right? So anything like a Zeal-Nielsen, which is used for TB, hematoxylin, Giemsa stain, different types of stains in histology are used to achieve different contrasts for different cell types. Finally, we have fluorescent staining. So there are a number of different dyes commercially available. As we said, this is not an exhaustive list. Different fluorescent proteins or conjugated antibodies, and you can label many different things with these with these kind of stains. I think we've got a lot of TB people who prepared these slides because there's a lot of tuberculosis examples here, but this is really can be applied to so many different organisms. So I encourage you to read the literature, look at what other people have done to best choose the way to achieve contrast in your sample types. And, you can achieve different things using different stains. And this is always how you should choose which experiment and which kind of contrast method you're going to use. So this is just a basic overview of how you would prepare your sample and fix it. So the first is really to not fix your sample at all. And these would be for kind of your very basic preparations. So something like a dry mount or a wet mount. Squash preparations and smears are usually not fixed. On the other side of this, where you need fixation, is usually with cells and tissues. So cells are present either as a cell suspension, and so you can do live cell imaging. Of course, these wouldn't be fixed because then you would be killing them and preserving them in that specific time and space. Otherwise, you can have adherent cell lines, and we'll talk about that in a minute. Or if you have a suspension that needs to be fixed onto a slide, you can cytocentrifuge them, and then these would be fixed. And then finally, of course, we have tissue samples that would be fixed in various ways. So this is also just something that needs to be taken into account. A lot of the times you actually don't need to fix the tissues depending on what you're working on. So let's look at different preparation options for all of these things that have been highlighted. This is really a basic overview. There's a lot to cover, and we can go much more in depth, and I'm happy to answer any questions after this presentation, and you can always contact me afterwards as well. So dry mounts really is the most basic preparations, and these are used for something like hair or feathers, pollen, dust, insect legs or antenna. I'm sure a lot of you guys would have done this in the first, we did this in biology. I think a lot of us would have done this, but essentially you position a specimen onto a slide, cover it with a cover slip, and put it onto the microscope. So this can also be done for small unicellular and multicellular organisms, or a membrane. I think some of us will remember doing the onion membrane, and can be stretched thinly onto a slide, and then you can image it. So very, very simple. And these can be stored for long periods of time, especially if they're dried samples, they won't go anywhere. So wet mounts are very similar. You would place a drop of fluid on the slide, whatever it is that your sample would naturally be in, put the specimen on top and then cover it. Fluid can be comprised of water, brine, glycerine, immersion oil, depending on the application or the sample that you're looking at. These cannot be used for a long period of time. Of course, the liquid will dehydrate and your living specimen is going to die. So something like a protozoa will live for about 30 minutes in that kind of environment. It's not going to be suitable for anything that's too large, like an amoeba, or anything that's moving really quickly. It's just going to move out of field rather fast. So you can put little strands of cotton in the water medium just to slow the movement down, but just bear in mind that anything too large is not going to be perfect for this kind of application. So squash mounts is really kind of looking into the intracellular components of cells. So here you're intentionally squashing or crushing your sample onto a slide and just looking at the content. So some like botanical specimens are beautiful for this. And you can look at like something like a chromosome. So essentially you would prepare a wet mount and then put another glass slide over the cover slide and just press down very gently to kind of squish and pop those cells. And then you can remove the excess water and just mount a cover slip over it. Smear preparations are slightly similar, but these would be done for samples such as blood, semen, cerebral spinal fluid, any screening for cervical cancer and scrapes, culture of microorganisms. So any pathologies that you would have seen where you've got a scraping or brushing or some sort of aspirate would be done for a smear preparation. And so for a smear, you essentially are preparing it almost like a wet mount with pipetting the liquid or your sample onto the slide. And then with another microscopy slide, you would use the edge and really smear and scrape it along, creating this thin and even coating along the first slide. After that, you can put a cover slip over the sample, remove any of the excess liquid, and then look at your preparation. So I would say for most of these, you should use an isotonic buffer. PBS works very well. And this is just something to take into account. Obviously, water will pop all your cells. Smears and squash preparations provide details about individual cells, relative cell numbers, but you're going to lose structural relationships, obviously, because you've burst open your cells or squish them. And these samples can be stored for a really lengthy period of time with no real special preservation methods. So let's talk a little bit about adherent cells. A lot of us in the field work on certain cell types, something like THP1s or cancer cell lines that you would grow in normal in vitro cultures can be prepared for microscopy really nicely. And so there's a simple way to do this is to use a cover slip that you would either sterilize. You can either just place it under UV, or I dip mine in ethanol, rinse them in water, and then put them in at the bottom of your well. And then you can seed your cells and just allow them to attach naturally onto the cover slip. And these will grow very well on uncoated glass slips. I haven't had any issues up till now, but if you're struggling, you can use cover slips that have been coated in polyol lysine or collagen just to attach them a little bit better. There are a number of commercially available chambers now that exist so that you don't actually have to fish out your cover slips after the fact. IBD and MapTech sell a whole bunch of these, but they are rather expensive. So if you're in a resource limited setting, I would say just stick with the cover slip method. It works really well. I haven't had any issues up till now. So what's really nice and convenient about this method is that everything is really present in this micro well format, and then you can do your fixation and your staining in this well without really worrying about anything running off or different slides that you have to move around. And only at the end, when you're ready to mount, you just fish out your cover slips. So this is a really convenient way. And so you can prepare lots of different conditions with stimulation or inhibition and do this kind of at scale. And this is a really convenient way to do this. So oftentimes you have a cell suspension and not cells that can be grown and attached. And so how do we get these onto a microscope slide so we can image them? So this is done using something known as a cytospin centrifuge or a cytocentrifuge. It is a specialized equipment, so you would need to have access to that, but a lot of the histology labs do have these present. And essentially, all that you're doing is you have this funnel system. I hope everyone can see let me just put this onto laser. You have the cytocentrifuge funnel that has this little hole here. It would be placed onto a microscope slide. And this little funnel here is where you would put your sample. We do this a lot in the immunology lab. It's a really nice way of looking at cells. So you attach this all together, and then it's placed in the cytocentrifuge. So essentially, it is a very low force centrifugation step. Anything higher than this will pop your cells. So you want to do this at very low speed, and it just concentrates the cells very nicely into a very small area, quite in a nice thin area. So if you've got the right cell number, you essentially are concentrating 20-fold your cells. So it's important not to put too many cells on, because then you're not going to have a one cell thick layer. So something like 20,000 to upwards of 100,000 cells per mil is usually a sweet spot for cytocentrifugation. And then these can just go on and be fixed like you would normally do for your adherent cells. So let's move on to fixation. We've now decided the sample type that we're going to use and which method we're going to go for. Fixation is really, really important. So as we've mentioned, preserving your sample in its lifelike state is really the key requirement here. So if you want to preserve your cellular structures, any proteins or nucleic acid to stain and detect them later, you have to fix them in their state. So you can also, there are a number of compounds that can achieve this. And if you don't want to commit to one, or you can't for whatever reason, sometimes you're out in the field and there's just no ways of accessing, say, paraformaldehyde, samples can also just be snap frozen at -80 degrees until you're ready to fix it in a specific way. And we'll talk about snap freezing as well, because there's some considerations there. So there are common fixatives, and there are other specialized fixatives you could use, but usually aldehydes, alcohols, and formulins make up the majority of the fixatives. So aldehydes work by cross-linking your protein and really do a great job of preserving cell morphology. I would say that's probably my preferred way of fixing, unless I have a specific reason why I wouldn't go this way. 4% paraformaldehyde is really great. It's usually 10 minutes at room temperature. So we have provided with kind of the standard methods of doing all of these procedures, but I encourage everyone to always optimize your samples. Sometimes you need either more or less, depending on the specific sample type, but these are good starting points for any experiment. With aldehydes, like paraformaldehyde and formalin, you can reduce antigenicity. So that's just something to take into account. If you're not detecting them, perhaps you need to do an antigen retrieval steps. And something with paraformaldehyde, I see a lot of people using 4% that's been stored in the cupboard for the last 10 years. Please prepare this fresh if possible, because your 4% PFA is probably not 4% anymore. So acetones or methanols also work very well as a fixative. They precipitate proteins, they maintain structures really well, especially cytoskeletal imaging. So if you're doing that kind of work, this is a really good way to fix the sample. If you have anything that is encoded, a GFP or anything like that, any genetically encoded fluorescent proteins, do not use any alcohol fixative, because this will dampen the fluorescence there. Methanol works very well for structure. Acetone permeabilizes as well and is a little bit less damaging than methanol, so just keep that in mind. And it's good to just try 100% as a starting point for 10 minutes, remembering that it needs to be ice cold. So usually we just put it in at -20 degrees. And then finally, formalin, which would be used for your standard FFPE samples, usually done in the histology labs. These can cause a high level of autofluorescence. Just bear that in mind. There are ways to dampen autofluorescence. I'm sure was touched on in the fluorescent protein lecture. Something like glycine can really help with that kind of thing. So for all of these fixatives, and I've mentioned this before, please use an isotonic buffer like PBS to make up any of your, so your PFA. It should be buffered so you don't pop your cells unnecessarily. Okay, so if you're looking at fixing cells, there are a number of ways of doing this. If you've just got hundreds of slides and you need to do this quickly, a Copland jar with a fixative inside when you've got lots of samples works extremely well, and it's nice and quick. If you've got cells that are in an Eppendorf tubes, you can just disperse them in the fixative, centrifuge them, and then do your wash steps. If you have something like a cut section or your cytocentrifuge cells, you can just pipette directly onto the specimen, and just using kind of that little bubble there to just stick onto the cells. If you're culturing your cells and it's very simple, just add it directly to your wells. And so this is kind of the stock standard protocol. You would immerse in, say, 4% PFA for 10 minutes at room temperature, usually 3 PBS wash steps for about 5 minutes. And then after that, if you're not ready to stain, your samples can be stored in PBS. And I would say really kind of long term. I've never had any issues. I've stored stuff for months after fixing at 4 degrees with no problems. But obviously it's ideal to stain kind of as soon as possible. So let's look at tissue samples. There's site considerations taken into account for that. Tissue sections, and thank you, Lisa, for providing this beautiful image here of a skin section. Tissues usually, generally, because of the need for them to be thin and transparent, need to be cut into thin slices. So usually we're looking at about 3 to 10 microns mounted onto microscope slides, although there are new techniques now where you can get thicker and thicker sections, but this is beyond the scope of this presentation. So most of your fresh tissues are really quite delicate. They move, they're easily distorted and damaged. So you need to prepare the sample specifically for it to be cut. And sectioning, it is technically demanding. It does need specialized equipment and specific expertise. So hopefully in your department, you have a histology area or a microscopy specialist that can help you with this. Lisa's the person we go to when we need some sectioning done, and there are different sectioning equipment that you would need for this. Now, the biggest thing when you're needing to section your tissue is the sample orientation, and I can't overstate this enough. If you, for example, in this section here, if you're interested in looking at this outer edge, if you had mounted the sample kind of the other way and just cut through the top, you wouldn't see this beautiful kind of layering here. So it's really important, don't just throw your sample kind of just willy-nilly into a chamber. You need to really think about what it is that you're looking at and how the sample needs to be orientated before you start cutting it. So in terms of preserving the tissue, as we mentioned, the tissues are very soft and delicate. So you can do it 2 ways. The first is to just snap freeze it. So you would use something like OCT medium. This is a water-soluble cryopreservation medium that's really kind of gentle, and it allows the cells to be kind of frozen very quickly and at very low temperatures without introducing any cellular damage because of, say, icicles or any of the low temperatures. And with that, so you can embed them in OCT and then store them really kind of very long term at -20 to -80. -80 is obviously ultimate if you really want to I mean, for years and years and years, but -20 works absolutely no problem if that's all you've got access to. The other option is to just do a fixed tissue preservation. So as we've mentioned, 4% PBS buffered PFA works very nicely, about 24 to 48 hours at room temperature. You can just pop it in your cupboard. And there are various reasons why you would choose either 24 or 48. So for example, when I have M. tuberculosis infected samples, we do them for 48 hours just to ensure that there's good permeation of the fixative and we've killed any infecting bacteria. Finally, if you're doing very standard H&E, or if you want to do formalin fixation, you would use PBS buffered 10% formalin. This is about 6 to 12 hours at room temperature. And this can be done usually in your histology lab. We'll have a big jar of 10% formalin. So these are the three kind of main methods of preserving your tissue. So for SNAP frozen tissue sectioning, you're going to want to maintain everything at a frozen temperature. And so to cut these tissues, we use something called a cryostat. And the important critical step here is that your tissue should stay frozen throughout the entire process, right, until you've finally done your fixing. So the sample obviously has only been frozen, hasn't been fixed. We haven't preserved any of the proteins. So you should take them out of the -80, maintain them in that until you can take them to the cryostat. So the cryostat can range between -15 to -35 degrees. This really depends on tissue type. And usually whoever's running the center there will be able to advise on that. And then you can mount them in cryomedia and cut at whatever thickness you want, and then mount them on your slides. So the slides can then be stored back in -20 or -80, or then you can immediately fix them and start your staining processes, depending on your kind of time availability. So post-sectioning, as I said, you can fix your tissues and then wash them as you would for your cells. So say 4% PBS, buffered paraformaldehyde, and then you can just store them at room temperature in PBS until you're ready to stain. So just a little note about stability. Cryosections from fresh tissues are superior in terms of preserving antigens and kind of cellular morphology or that kind of thing. But FFPE sections are going to be much more stable for much longer. You can store your FFPEs for, many years without anything kind of happening to them. But just know that your antigens are probably not going to be as available. And so an antigen retrieval step is usually included with FFPE sections. So if you have a PFA fixed tissue preparation, you would have fixed your tissue in PFA. We're usually looking at about 5 to 10 times the tissue volume. And this is just dependent on what you're working with, but just bear this in mind, you need enough fixative to permeate your tissue. And then what you can do is embed the tissue in something like a low melting point agarose. And depending on what you're using to cut, 2 to 4% low melting point agarose works very well. And then section your tissue to the desired thickness. So you can either use a microtome if you're going for very, very thin sections, or a vibrotome if you're going for something a bit larger, like 500 microns. And then once you've cut your tissue, you can mount your sections onto microscope slides. Now they've already been fixed, and these are kind of ready now for some staining. So FFPE tissue sample preparation is slightly different, but kind of in the same realm. Orientate your specimen into a little cassette. And this is then covered in warmed wax. And then the block is just allowed to solidify on just a cold surface. And then you can remove it for your mold and it's ready for sectioning. So again, please remember the orientation is really important. So how you put it into the cassette will really determine what you're able to visualize after you cut. So once it's in the cassette, that's kind of it. So really think about that when you place your sample to ensure that you're getting the correct orientation and access to what it is that you want to visualize. And as we've mentioned, FFPE samples usually require antigen retrieval, but these can be stored. They're very, very, very stable for years and years and years. In fact, we're busy looking at samples that were archived about 40 to 50 years ago, and they look absolutely brilliant. We did have to retrieve some antigens, but you really get kind of very long-term stability with FFPE. So if you're running a multi-year kind of study, this is kind of the way to go, I think, in terms of tissue preparation. All right, so let's talk a little bit about permeabilisation. We've now fixed our samples. Everything is kind of in its lifelike state where we want it to be. And now we need to kind of access whatever it is we want to image. So permeabilization is really important, especially if your target is intracellular. So if your antigen is something that's a subcellular, you're going to need to punch holes into the membrane so that whatever it is that you're using can enter the cell. Specifically antibodies, which are quite massive, they need to enter the cell, so you're going to need to permeabilize your cells. Methanol and acetone fixed samples have already been permeabilized. So if that's what you're using, please keep in mind, you do not need a permeabilization steps. And just a note on methanol, acetone versus paraformaldehyde, each one of these fixation steps can introduce mild aberration in your samples. And so I encourage you to just kind of read around that and what that can kind of cause. Sometimes you get a little bit of protein precipitation with methanol or acetone. So if you're looking at a specific protein, just keep that in mind that perhaps PFA might be better in this instance. So like I said, different reagents, times, concentration, they will give you different results, and you need to optimize that. In fact, I would say if you've got enough sample, test the different fixation and permeabilization methods and just look at the different results. And it can actually be quite jarring how different permeabilization and fixation can introduce completely different sample quality. And your specific sample will need a very customized method. So please do this, please optimize for each and every sample type that you use. So if you're doing a multiplex experiment where you've got membrane-bound and intracellular targets, this is quite common. If you need to visualize both at the same time, you should stain your membrane-bound antigen first, then permeabilize, and then do your intracellular staining. So that's the procedure you would take for that. If you do your permeabilization first, your membranes will be so destroyed that you will not have access to any of those targets. So there are a number of detergents and can be used for permeabilization. Triton X100 really probably is the most popular detergent, really good penetration for antibodies. You would incubate your samples 0.1 to 0.25%, depending on the sample type. Usually start with the lowest and then work your way up. You don't want to over permeabilize your sample, and this is usually done for 10 minutes. And then it's very important to wash with PBS after that. It's, as mentioned, sometimes it's not appropriate for membrane-associated antigens, especially Triton's destroys membranes quite readily. So you should just determine the percentage, and if that doesn't work, there are other detergents that are available. Something like NP40, Tween 20, or Saponin are very common and achieve the same kind of permeabilization. So we have now permeabilize our cells. We're ready to label. Which label strategy do we use? So for H&E, we wouldn't have done any permeabilization. H&E staining really gives you good contrast. It's a nice dye. And so although we have natural pigments in our cells or in the tissue, usually specimens are quite colorless. So if we want to look at bright field microscopy, you need to do some sort of staining. H&E is really the very universal, well-standardized stain to do this. And if you have a histology department, they will be able to run you through the protocol quite easily. So in histology, it's really kind of the stock standard workhorse of staining, and it's very quick and inexpensive. So hematoxylin specifically stains the nuclei, this kind of purplish blue color, and then eosin stains the extracellular matrix and cytoplasm pink. And then you have different structures or kind of different shades and hues. And so a combination of these colors gives you this really beautiful contrast. And so something like when you're doing a differential cell counts, you can identify T cells and macrophages and different immunological cells just based on the kind of shades and hues that they have. So definitely look at examples in the literature, and there's some very nice protocols and manuals so that you can really have a good identification of your different cell types. So H&E is really a good stock standard to start with if you're looking at kind of your basic histology. So this is just a very quick procedure. It does get a bit more complex, but basically the first step is to just dissolve all the wax away, right? Remember you had your melted wax, and we do this with xylene. You then need to remove the xylene, and this is done through several changes and washes through alcohol. Then you would do your nuclear staining, for example, with hematoxylin. That's just the stock standard one you would use. And then you blue the sample with a weak alkaline solution. After that, you can just remove your excess background with a very weak acid and alcohol mix. And then finally, you're ready to counterstain non-nuclear components with the eosin, as we mentioned. And then you would rinse, dehydrate, clear, and then mount with a cover glass so that you can visualize onto the microscope. So now let's move on to fluorescent labeling. This was already touched on the microscopy segment, so I'm sorry if there's a bit of repetition. And fluorescent labeling already has been kind of this very powerful tool in cell biology and microscopy. And this is because of the advent of fluorochromes, which have made these multicolor experiments possible. These fluorochromes are very sensitive, very stable, and don't really interfere with function of target molecules. You can do live cell imaging. And so there's two ways to do this. The first is with genetically encoded fluorescent proteins, and you can do live cell or fixed cells, or you can use fluorescently labeled antibodies, which is why we call it immunofluorescence, because we use immunoglobulin and fluorescence to label different antigens. And so I apologize for the repetition here, but essentially antibodies for immunofluorescence are made up of these large immunoglobulin molecules, and these recognize antigens via the Fab's variable regions. And this is this kind of lock and key mechanism that can recognize antigens. And so the epitope is the part of the antigen molecule to which the antibody attaches itself. And so new antibodies that are coming out now, that are conjugated antibodies now, are even smaller. You can buy just the Fab fragments, especially when you've got something like you can't get the antibody in. Look at getting kind of the Fab fragment antibodies that work really nicely. I've been using that at the moment, and they're much smaller than these kind of large immunoglobulin. So just a touch on polyclonal versus monoclonal. I have students ask me this often. And I'm an immunologist, so I couldn't resist putting this definition in. But if you're looking at a polyclonal antibody, basically the antibodies have been made from different B cells. So these will recognize multiple epitopes on the same antigens. And so this is nice, for example, if you've got for example, my PhD, I worked on abalone, and there are no commercially available abalone antibodies. So we used a polyclonal antibody from different mollusks just to increase kind of the sensitivity, but you obviously decrease your specificity because it can recognize multiple epitopes. There's a bit more non-specific binding. If you're working with human samples or rats or mice or anything like that, monoclonal antibodies are made from its single B cell, and they will only bind to one unique epitope. So you've got increased specificity, but decreased sensitivity. But honestly, the antibodies nowadays are just so well characterized that this isn't really kind of a major problem. And I would say if you can get a monoclonal antibody, go for that. So you can either go for a direct versus indirect labeling strategy. So your primary antibodies can either be already conjugated to your fluorophore. So this is called direct labeling. So you would have a single antibody binding and a single fluorophore. Or you can go for an indirect labeling where you have one primary antibody and then secondary antibodies with their conjugate on it. And so multiple secondary antibodies can bind to a single antibody. So if you've got a really rare cell population or something that's very lowly abundant, I would say go for your indirect strategy, because then you boost your signal. It's just important to remember that your secondary antibody then may bind non-specifically, but that's why we have controls and we take that into account. The big thing to remember here, and you guys will probably already know this, but your secondary antibody must be raised against the host species of the primary, not your antigen. So if this is a mouse sample, this is an anti-mouse primary antibody raised in goat, your secondary antibody needs to be an anti-goat secondary antibody. All antibodies should be optimized. So you should do I usually use recommended concentration a little bit lower and a bit higher. And so do a titration. And then in your controls, and I'll talk on this later, we must include secondary only controls if you've got a secondary antibody to ensure that any signal that's being generated is not because of non-specific binding, but because your secondary antibody is binding specifically to your primary. And of course, an unlabeled control to ensure that any background autofluorescence signal is already taken into account. So blocking in immunofluorescence is extremely important. You have to block the samples in order to reduce non-specific binding. So non-specific binding happens for a number of reasons. Unreactive fixative aldehydes, these can cross-link antibodies to inappropriate structures, causing non-specific binding. There are samples, depending on your sample type, that can trap antibodies. And so sometimes just putting a little bit of tween really helps with that. And then, as we mentioned, polyclonal antibodies, you have these low affinity molecules, so they can bind kind of non-specifically. So blocking will aid with that. Usually we treat with a protein solution. And this can be the stock standard ones, really bovine serum albumin, your BSA, or casein, like just the normal milk powder that you would buy in the shop works also quite nicely. If you don't have access to BSA, sometimes BSA is not available in the for various reasons, for import reasons. Gelatin can work. The ultimate blocking buffer is usually the serum that is made from the same species to which the secondary antibody is made. So if you're using an say like raised in goat, you would need a goat serum, but that's not always available. And I would say in resource limited labs, we usually just use BSA or milk powder. So please don't forget your blocking buffer, and it's usually done for an hour at room temperature, and then you would make up your primary antibody in a blocking buffer, the same blocking buffer. After you've labeled your targets, usually we use a counter stain very similar to the different dyes. The stock standard is to label your nucleus, usually using DAPI or Hurst. These are really nice counter stains, and it's very bright, fluoresces very brightly. It binds to the minor groove of double-stranded DNA quite strongly. It's also nice to label the cytoskeleton, just to look at cell boundary borders. And to do this, we use phalloidin. And this is a cyclic peptide that comes from the death cap mushrooms, Amanita phalloides, which I find so interesting. And this has very, very high affinity towards filamentous actin specifically. And then you can see these beautiful filaments. And this is another image by Lisa, which really kind of shows the stunning cytoskeletal stain and nuclear stain. So we've talked quickly about the controls. Why do we need fluorescent controls? And please always include these. We need to make sure that you provide evidence that the staining that you have is real and not just due to some autofluorescence or non-specific staining. And if you want to get your work published, this will be asked to be provided in your supplementary information. And this helps to make your data interpretation easier, narrow down any issues that you're having. So if you're not getting a signal, at least with your controls, if you've got your positive control, you know that it's not just because something happened during the staining procedure. So the type of controls I would recommend is an unlabeled sample, especially with tissue, you're going to get a large level of autofluorescence, especially if you use something like formalin or paraformaldehyde. So you want to determine that background fluorescence and then take that into account. If you're going for primary, secondary approach, you would have a secondary antibody only control. So if your secondary antibody brings any sort of staining, you know that you've got unspecific binding occurring, and then you need to take that into account, like including tween in your blocking buffer and in your wash steps to try and get rid of some of that non-specific binding. A negative control, so this isn't always possible, but ideally, if you have a sample where your target is not expressed or you can block it, so yeah, for example, an unstimulated sample, this is a really nice one to have to show that your target doesn't just come up randomly. And then a positive control, also kind of difficult to get sometimes, but a sample, you know, the target's expressed so that you can ensure, okay, my antibody is binding to this target and I know it's there. But this isn't always possible, but unlabeled samples and secondary antibody controls should always be included. So finally, we've done our fluorescent staining and washing. We're ready for mounting and imaging. So you need to seal your sample and preserve it for long-term storage. Usually your mounting media will do a lot of things at once. They have agents that scavenge free radicals, preserves the samples, raises the refractive index so that it's appropriate for your microscope, and reduces photo bleaching. When you do a mounting media, something like DACO, you should allow the slide to kind of cure overnight at room temp. Don't stick them straight in the fridge. They won't cure and won't dry. So just let them dry in the cupboard in the dark overnight, and then you can do your final sealing. And then slides, all your slides, especially if you've got fluorescence, should be stored in a light, tight container to prevent any photo bleaching. And if you're storing them at 4 degrees, you can really store them for months, if not years, I would say, if they're kept in the dark. I've imaged samples years later with absolutely no loss in fluorescence. So just to summarize, I hope you guys have kind of a new appreciation or a revived appreciation for sample preparation and why it is important. There are a number of things that you need to take into account, and please plan this before you start acquiring your samples. I've been approached by people who are working in the field on on very important sample types, and they just fix them completely wrong. And it's so hard to then go back and change things. Or if you want to look at specific targets and you've fixed it inappropriately, you're not going to be able to look at that. So please, it's important to take all of these things into consideration and just have a good plan of action from the get-go, so that you have the best and optimal quality samples that will give you the best results. With that, I just want to thank you guys all for your attention. And I want to thank the ABIC team for putting this all together, especially for kind of editing my slides. I did provide the content, but they made it all look beautiful with animations and extra information. So thank you to my colleagues at ABIC, and it's just been an absolute pleasure to work with you.